Hello, and thank you for your interest in our study, where we showed that knockdown of splicing complex protein PCBP2 reduces extravillous trophoblast differentiation through transcript switching. Let me guide you through our poster with a quick background story and highlights of our projects. Mutations in the link help non-coding RNA, also known as HELPR, have been associated with familial forms of the pregnancy-specific HELP syndrome. These mutations negatively affect extravillous trophoblast differentiation from a proliferative to an invasive state, and also disturb the binding of RNA splicing complex proteins, PCBP1, PCBP2, and YBX1, to link help non-coding RNA. In this study, by using both in vitro and ex vivo experiments, we investigated the role of the RNA splicing proteins YBX1, PCBP1, and PCBP2 in the regulation of in vitro invasion during first trimester placenta development. Additionally, we study if this regulation is due to alternative mRNA splicing. By using a manufactured extravillous trophoblast cell line called HCR8 SV neo cells, and also human first trimester placenta explant, we investigated the effect of sRNA-mediated downregulation of PCBP1 and 2 and YBX1 genes on the differentiation of EVTs. Translation assays that we see in the graph on the left and proliferation assays that we see in the graph on the right indicated that upon knockdown of PCBP2 and to a lesser extent YBX1 and PCBP1, EVTs fail to differentiate toward an invasive phenotype. The same pattern was observed in placental explant, where PCBP2 knockdown led to approximately 80% reduction in the number of explants, showing an EVT outgrowth. Here we see representative immunohistochemical stainings on proteins phospho-H3 that indicates cells that undergo mitosis, and SLHC protein that indicates the location of EVTs. After the quantification of these immunohistochemical stainings and calculating the ratio of H3 to HLA-G, we saw that there is a significant induction of proliferative EVTs upon PCBP2 knockdown, while YBX1 silencing leads to a reduction of proliferative EVTs. To further investigate the effect of PCBP2 silencing on EVTs, we performed whole transcriptome sequencing on HCR8 SV neo cells after PCBP2 knockdown. Then we saw that PCBP2 silencing led to a switch of splicing of 310 genes with predominant function in cellular assembly and organization, cellular function and maintenance, cellular growth and proliferation, and the cell cycle. Something that is very interesting, as EVTs, upon differentiation, alter their functions to be able to invade the city of the mother by changing the cellular assembly and their proliferative activity by exiting the cell cycle. To further validate this analysis, we chose 3 genes. 2 genes that showed exon exclusion upon PCBP2 knockdown, which are the NCOR2 and EXOC7, and 1 gene showing inclusion, which is the FLMB. And for each gene, we designed primers recognizing the included or excluded exon, and a set of primers recognizing all transcript of the specific gene expressed in HCR8 cells, and we performed qPCR analysis. So as you can see here, we were able to validate differentiation splicing upon PCBP2 knockdown in all the 3 genes that we chose. Finally, in our study, we identified a C-rich motif that is specifically located adjacent to exons that are excluded upon PCBP2 silencing. In more details, we constructed a data file containing all intronic regions affected by PCBP2 knockdown that are directly adjacent to the significantly excluded exons of the differentially spliced genes after PCBP2 knockdown. To this data file, we also added all intronic regions adjacent to included exon upon PCBP2 knockdown, and also added the 5 and 3 end regions surrounding transcription start sites that were excluded upon PCBP2 knockdown. This data file was submitted to meme motif enrichment analysis using a zero-order background model. And this provided us with a highly significant motif that is shown in this picture. So this motif was found in 75% of genes in regions with 100 base pairs from the exon boundaries of exon excluded upon PCBP2 silencing. The adjacent region of excluding transcription start sites did not contain this motif, while the regions adjacent to the included exon of one of the two genes contained this motif. So gathering all this data, we can say that PCBP2 through mRNA splicing appears to be a paramoleculator of extravillous Trophoblast MET differentiation, where its disturbed binding to lin help could contribute to dysfunctional placenta development, as seen in the HELP syndrome. Thank you again for your interest in our study, and in case of any question or suggestion, please don't hesitate to drop me an email.